Guess it's about time to. So welcome back to our half of the program session is Kaitlin Epperson. And the
All right. Thank you. Hear me in the back? Okay. Okay, awesome. Thank you so much for the opportunity to speak with you all today. Before getting started, I'd like to make special mention of my lab mates, both current and previous, that have made this work possible, especially Dr. Perry for all of his time and effort. And so I would like to talk with you today about how the development and function of the bovine corpus luteum is mediated by vaccination at AI. And so, as I'm sure most of you all are very aware, there are several different infectious reproductive diseases that are of particular concern for the beef breeding herd. And among the different diseases, these three agents are what we'll be concerned about today, all viral agents. And we're also aware that vaccination can serve as an important tool for implementing herd health measurements, but also in protecting our females from succumbing to these diseases. But we're also aware that there are negative consequences for reproduction related to the timing that vaccines are applied. And so oftentimes we may get producers that ask us, why not vaccinate for pre-breeding diseases during the time of synchronization while my cattle are already in the chute? And so to answer this question of whether or not this would be plausible, Dr. Perry in 2013 had naive heifers that were vaccinated with a modified live at the time of induced ovulation, so you can see in the protocol pictured to the right. And what he discovered was that there were increased percentages of abnormal estrus cycles, and in particular, we see a 38% of abnormal estrus cycles in that modified live group compared to animals that had received the killed vaccines at 10 and 14%. And 0% in the control. Also, in terms of pregnancy success, we saw that there was decreased pregnancy rates. For the modified live group, they had a 33% pregnancy rate following the synchronization period, whereas the control was at 90%. And then lastly, there was also disrupted progesterone and estradiol profiles, illustrating that there's clearly some type of mechanism that these viral agents are causing after vaccination during this time of a synchronization protocol. And so moving forward, the next question was, what if we vaccinate according to per label instructions, which requires you to vaccinate 30 days before breeding? And so this time, Dr. Perry in 2016 used a little over 1,400 cows and heifers that had already seen vaccination before. And again, this time we see that the modified live treatment group had decreased pregnancy rates at all time points measured. Specifically, AI pregnancy rates were reduced by 6.5%. At day 56, they were reduced by a little over 4%. And then for the breeding season in total, they were still reduced by almost 3%. And so clearly we're seeing here that there's some type of mechanism occurring that even when modified lives are applied according to per label instructions, there's something happening that is affecting the bovine reproductive system. And so, as I mentioned, there's a handful of different negative consequences that can happen following native infection of these viruses, but also following vaccination. And the focuses of today will be why exactly these things are occurring after we apply pre-breeding vaccinations. And what you'll notice from the data I'll present today is that collectively it indicates that we're getting some type of ovarian dysfunction occurring. And so the first question being is, what type of dysfunction is occurring and how might that be mediating pregnancy rates and reproductive function? And so in 2016, again, Dr. Perry had collected a handful of cows that were estrus synchronized and then vaccinated with two types of a modified live, and then also with a killed vaccine. And they also collected at days 15 and 16, and then later at day 30, a handful of different tissues to be analyzed for virus isolation. And so you'll notice that of all the tissues that were analyzed, which included granulosa cells, follicular fluid, uterine fluid, and endometrium, all were negative for presence of those 3 viruses that are commonly included within pre-breeding vaccines. But what's very interesting is when looking at the different titers among the 3 viruses, we see some peculiar results. And so I'll point out that the blue and orange bars represent the 2 modified live treatments, whereas the orange yellow represents animals that had subluteal progesterone concentrations when we would have expected. Here is that the 2 titer concentrations, we see that from day 0 to day 15, there was a significant increase, which is somewhat different than where they either stay the same or they indicate to us that we're having some type of mechanism that's not necessarily through direct infection. And so the current goal of our ongoing research then is to kind of analyze what specifically was occurring that might have caused the subluteal concentrations. And so we wanted to evaluate the influence of vaccinating with either a modified live or a killed at the time of AI on Corpus Luteum development and its function. 9 beef synchronize them with the 7-day co-sync protocol started. We came in and we vaccinated these cows with either a modified live or a killed vaccine, and we also ultrasounded to identify dominant follicles. And we returned on day 3 to ultrasound again to confirm the disappearance of those follicles in hopes of that they had ovulated and developed a corpus luteum. On day 10 through 12, we collected a subset of 13 females that were subjected to ovariectomies, and we also had 10 extra controls that did not receive vaccine that were also ovariectomized. And when we collected ovaries, we dissected out the corpus lutea so that they could be prepared for H&E staining for histology analysis. And I'll also mention at all time points that have a blood tube indicated that we had collected plasma for analyzing progesterone and cytokine concentrations. And so really briefly, the stats that we used for this was in SAS with the mixed and GLM procedures, and significance as always was less than or equal to 0.05. And first, I'd like to point out some of our histology results, and I'll remind you what we typically would expect to see from a normal corpus luteum H&E stain. And so you can see here that the large luteal cells are these large, puffy, circular appearing cells with round nuclei and abundant cytoplasm, whereas the small luteal cells appear similarly but much smaller. And so if we use this as a comparison point and look at what happened for the killed and inactivated treatments, you'll immediately notice that there's a greater number of cells present here that are likely not luteal cells. We can't tell for sure, but we suspect they may be plasma or fibroblast cells. Also, in stark contrast, when looking at the modified live histology, you'll notice there's abnormally few number of cells in panel C, and the cells that are present don't appear to be healthy. They have very flattened, oddly shaped nuclei and very little cytoplasm. And similarly, with another example of our modified live, this has an intermediate number of cells here, but I would like to point out these cells here with the white-looking cytoplasm, which I suspect are apoptotic. And so to quantify this data, myself and Lacey Quayle in the audience, we counted the total number of cells in these pictures and then as well as the large luteal cell counts. And so what you'll notice is that this picture showing the killed group was indeed indicative of higher total numbers of cell counts, which again, I suspect are probably not luteal cells here, probably something else that had invaded at that time. And I'll also mention that the 2 modified live bars that are shown are split between a new CL and an old, which simply describes the animals that had 2 corpus lutea at the time of collection relative to when they had ovulated. And then lastly, for the percentage of large luteal cells, you'll see that our modified live treatment had the lowest number of large luteal cells compared to the killed being intermediate and the control at the highest. And so moving forward, what we wanted to progress with this data is, now that we've seen structural and morphological differences in the corpus luteum, does this equate to functionality? And the first thing we wanted to look at was if we had presence of abnormal estrous cycles determined by progesterone profiles. And as you can clearly see, there's a 24% incidence of abnormal cycles for the modified live group compared to 0% in the killed. But if we break this down even further, a good portion of that 24% had a corpus luteum that regressed before day 16, and then the remainder had a delayed rise in progesterone after they had ovulated. And so it's not surprising that, considering this data as well as the histology results, that when we looked at the treatment by time effect on progesterone, there was clearly a decrease in our modified live treatment, shown in the blue bars during the different time points measured. So thus far, what this data is implying to us is that there are significant influences on corpus luteum morphology as well as its ability to produce progesterone, but we're still questioning at this point, what are the mechanisms that might be causing this damage? Because these are modified live vaccines, and as we had shown before, they're not necessarily directly invading the tissues. And so to try and answer that question, we wanted to evaluate more of the immune side of things. And so we took our plasma samples and subjected them to a multiplex cytokine analysis where we evaluated 15 different cytokines shown in the far left-hand portion of the table. And I won't belabor each of these cytokines, but I would like to point out that the first 2, interferon gamma and macrophage inflammatory protein 1 beta, were the 2 that had significant treatment effects. And what I'd really like to illustrate, though, are these 2 cytokines with their treatment by time interactions. And so, firstly, interferon gamma, and I'll also mention, too, that for all graphs that I show you today, the orange line represents the killed treatment, the gray line is the modified live who had abnormal cycles, and then blue being modified live with normal cycles. And so you can see in the interferon gamma treatment by time that we had a significant rise on day 2, which is not overly surprising considering it's a pro-inflammatory cytokine, and this is 2 days after vaccination. But what's very interesting here is that we had a significant rise in our modified live treatments compared to the killed. And then following for IP10, or interferon gamma inducible protein, we also saw a similar spike for the modified live on day 2 compared to the killed treatment. But what's interesting here is that before all of these concentrations fell and remained constant on day 6 to 22, you'll notice that between days 2 and 6, the killed treatment had a rise in this cytokine. And like IP10, monocyte chemoattractant protein 1, you'll also notice that the only significant differences in this graph is here between the 2 to 6 mark of the killed vaccine. So this is somewhat mimicking what occurred with the IP10. And then lastly, and maybe most interesting, is the macrophage inflammatory protein 1 beta. And here what I'd like to highlight is that the modified live abnormal treatment had greater concentrations throughout the sampling period compared to the other 2 treatment groups. And so to summarize this data for you, what we've realized is that modified live vaccination does clearly negatively influence the ability of the corpus luteum to either develop and persist, as well as produce progesterone. And we're also realizing that there's different cytokine profiles that might indicate that there's a stark immune response occurring that might be mediating some of these effects. And as you could see from the 4 different cytokines I presented today, there were increased concentrations from a modified live being administered. And then lastly, I'd like to mention we will be doing some future work to try and identify what leukocytes might be involved in this process to hammer out what exactly is occurring for the corpus luteum after vaccination. And so with those, I would be happy to take any questions you might have. Yeah, Dr. Brazier?
Did you look at any of the type 1 interferons like alpha?
Yeah, we did. We had a lot of interferon, actually a handful, I think. No, I'm sorry, I'm confusing with the interleukins. We looked at a lot of interleukins, but I don't believe we looked at any other interferons. Gamma was the only one, but that would be interesting.
I just noticed that IL-10 was up, seems to be higher than one better.
Mm-hmm. It's very interesting because IP10 as well as MCP1, I've read some stuff that a lot has been published in PCOS in women, and those have had increased concentrations as well for women that exhibit that. So it's very peculiar.
Kaitlin, have you checked into the Corpus Luteum concept of gene expression of cytokine?
We have not. That's something I'd really like to do, budget willing, but that would be something that I was really interested in doing. That's a great question. The question was, are there other tissues that are being negatively influenced, such as the corpus luteum, right? Yeah, I can't honestly answer that very confidently for you because I've been mostly focusing just on this. But I would say that I would suspect that if I had to guess, there's probably gastrointestinal things that can occur, because especially BVD, that is a virus that has an affinity for areas of the gut, like the jejunum. So if I had to speculate, I would guess yes, there's probably other tissues that are showing similar types of short-term damage. Okay, thank you.
You know, okay. Dr. Baser. The title of his talk is The Ovine Utero Placental Development.
Thank you, Dr. And I want to thank the IFRB committee for providing me opportunity to present at this platform today. So I'm going to talk about creatine metabolism in uterine and conceptuous tissues. So we have all seen this slide this morning in Dr. Beza's presentation, but I want to go through this slide so that we are all on the same page. So creatine, we all know that creatine, the importance of creatine metabolism in homeostasis of ATP in tissues such as the brain and muscles. So creatine, the source of creatine in the body can be through the diet or through endogenous synthesis. Creatine is synthesized in the body by the subsequent action of two enzymes, that is AGAT, arginine glycine amidinotransferase, and GAMT, guanidino acetate methyl transferases in the liver. So first, guanidino acetate is synthesized by the action of AGAT in the kidney, which is transported by transporter SLC6A13 in the liver, where it is methylated to form creatine. And this creatine is then transported to target tissues such as the muscles and brain by the transporter SLC6A8. And in target tissues, creatine is then converted to phosphocreatine and back to creatine for the regeneration of ATP by the enzyme creatine kinase. However, the muscles and brain are not the only tissue that requires high amount of ATP. The conceptuses also require high amounts of ATP. In ruminants, the conceptus during early pregnancy hatches out, elongates, and then followed by apposition and adhesion for implantation. And this elongating conceptus is highly proliferative, as shown here by the PCNA staining in the implanting conceptuses. And when we look more histologically, we can see that during this implantation stage, the mononuclear trophectoderm cells divide and they form trophoblast giant cells, and these giant cells invade into the uterine luminal epithelium, phagocytize those cells, and eventually form syncytial plaques. So all of these events require huge amount of ATP. So we hypothesize that the ovine conceptuses and uterine luminal epithelium and glandular epithelium cells has the synthetic machinery for the synthesis of creatine and phosphocreatine, and also their transporters. To test our hypothesis, we use the sheep as the model animal. So on day 0, when detected in heat, the ewes were bred to fertile rams, and tissues were collected during early pregnancies and late gestation, especially uterine tissues on day 9, 12, and 17, and also on day 125 of pregnancy, and conceptus tissues on day 15, 16, 18, and 20, and also placental tissues on day 125 of gestation. And those tissues were then processed for immunofluorescence staining to determine the localization of the enzymes and transporters in the creatine metabolism pathway. So, and here are the results of the immunofluorescence staining. The top panel here shows the localization of the enzyme AGAT that synthesizes guanidino acetate, and then the bottom panel here shows the transporter for guanidino acetate. As we can see here, by immunofluorescence staining, the AGAT was not detected during pre-implantation or the peri-implantation period. However, using the same antibody, AGAT was localized in the proximal tubules of the kidney. But interestingly though, there was expression of guanidino acetate transporter in the conceptus trophectoderm cells during the peri-implantation, as well as during the pre-implantation, as well as during peri-implantation period. And there was higher expression of this transporter during the peri-implantation period. So what happens to the guanidino acetate? Is it utilized to synthesize creatine or not? So yes, it is. The top panel here shows the localization of enzyme GAMT for creatine synthesis and the expression of SLC6A8, that is the transporter of creatine. And this is, so we can see here GMT was localized in the conceptus trophectoderm cells during pre-implantation and during the peri-implantation with higher expression during this period. However, there was higher expression of this transporter for creatine during pre-implantation compared to the peri-implantation period. So higher expression of transporters during pre-implantation and higher expression of the enzyme during the peri-implantation. So how is this creatine used by those conceptus tissues? So the answer is creatine kinases, and the top panel here shows the brain type creatine kinase CKB expression, and the bottom panel here shows the localization of the muscle type creatine kinase and the mitochondrial isoform of creatine kinase. As we can see here, the expression of CKB was limited in trophectoderm cells during pre-implantation. However, there is a higher expression of these creatine kinases on days 18 and 20, and similarly high abundance of the muscle type and the mitochondrial isoform of creatine kinases, as shown here in the model figure. Now I'm going to show the results for the endometrial tissues. The top panel again here shows AGAT, the enzyme for GA synthesis, and the bottom panel here shows the SLC6A13 transporter for GA transporter, and this is the IgG control here. So again, AGAT was not detected in the endometrial luminal or glandular epithelium tissue, but using the same antibody, it was localized in the proximal tubules of the kidney. And interestingly, also again, in the endometrial tissues, the transporter for guanidinoid acetate was localized in luminal epithelium, also in glandular epithelium, and in the blood vessels here during pre and the peri-implantation stages. So even though they do not have the enzyme to synthesize GA, GA is probably transported from the maternal vasculatures in the endometrial tissues. And this here shows the top panel again shows the enzyme GAMT for creatine synthesis, and these are the IgG control, and the bottom panel shows for the transporter of creatine. So GAMT, the enzyme GMT was localized in the luminal epithelial cells and also in glandular epithelial cells during both pre and peri-implantation period, and the transporter for creatine was not detected or had very lower expression during the pre-implantation. However, there was abundant expression during the peri-implantation period. And this is the creatine and kinases in the endometrial tissues. The right panel here shows the IgG control for the antibody for creatine kinase, the brain type creatine kinase, the muscle type creatine kinases, and the mitochondrial type creatine kinases. So very interestingly, we can see here all the three isoforms of creatine kinases had higher expression in the luminal and glandular epithelial during the pre-implantation period. However, not as much, and like a lower level, not almost undetectable CKB in the luminal epithelial, and similarly these two isoforms had also comparatively lower expression. So what does this all mean? To summarize this, I have two model figures here. First for the pre-implantation and the next for the peri-implantation. So in contrast to our initial hypothesis, neither the uterine tissues nor the conceptus tissues have the enzyme AGAT for the synthesis of guanidinoid acetate. However, they do have the transporter SLC6A13. So likely the guanidinoid acetate is transported from maternal vasculature into the endometrial tissues and further into the conceptus tissue, and both the uterine and conceptus tissues have the enzyme GMT and creatine kinase for the creatine phosphocreatine pathway. So, and interestingly, there was temporal differences. So initially during the pre-implantation period, there is higher expression of proteins of the creatine kinases. However, there is higher expression of creatine transporter during the peri-implantation. And in conceptus tissues, before implantation, there is higher expression for creatine transport. However, there is higher expression during the peri-implantation for GA transporter and the enzyme for creatine synthesis and also the creatine kinases. So meaning potentially at this stage, the conceptus is actively using this creatine and phosphocreatine shuttle to regenerate ATP for all these implantation events. And today I do not have the time to show the day 125 data. However, we found that there is expression of the enzyme AGAT in the intercarancular epithelium in late gestation, especially day 125. So meaning that arginine and glycine from the maternal vasculature can be transported in this tissue, and which can synthesize guanidinium acetate and further transport it into the chorionic epithelium, the fetal epithelium, and also creatine can be transported directly across the carancular stroma into the chorionic epithelium, and the chorionic epithelium has the enzyme GMT and creatine kinase. So the placental tissues can use this creatine and phosphocreatine shuttle for the regeneration of ATP. So what are the implications? Why is this creatine then important? So the creatine can be used for the regeneration of ATP to further support cellular proliferation, cell migration, cytoskeletal remodeling during the peri-implantation period, and also in late gestation for the fetuses. It can help in myogenesis and neurological development for enhanced fetal qualities. With that, I would like to thank my PhD mentor, Dr. Fuller-Bezer, and my PhD advisors, Dr. Johnson, Dr. Wu, Dr. Burkhardt, and I would also like to extend my thanks to Dr. Claire Stenhouse, Dr. Huan Seo, Catherine, Robin, Avery, and Bran for their help and support. And I thank you all and take any questions now. Dr. Wells got one question.
I like the implication slide that you just showed. It made me think about the muscle section that you showed. Primary and secondary.
18. We do not have any data currently for the got keep in the loop. We will be posted here. Thank you. Sorry, I don't think I get your question.
Well, you have a shallow GP will express genes the same around. And I saw a lot of your slides where it looked like
That's a good question. I'm not sure, but I think, yeah, I don't know. I'm not sure.
really interesting.
Yeah.
So were you having
So this is implications. There is not our data, but data from other rat studies and other species. They have seen increased expression in muscles actually. So myogenesis. So especially regeneration of ATP for when there's mesenchymal cells differentiate into muscles forming cells. So that's what they are stimulating.
Okay, our next speaker is Brianna Meyer from Dr. Duncan McKenzie's lab. And the title of her talk is A Novel Approach to a New Question, Evaluating Ecological Breeding Strategies in Sea Turtles.
All right. Hi, everybody. And now for something a little different. I want to start out by talking a little bit about a species that you may not have thought about recently. And we're going to start just with a general discussion of the importance of reproduction, which I'm sure everyone here has some thoughts on. So a species' success is dependent on its reproducing adults making the best choices. And making a wrong decision about when to feed or when to breed can easily ruin a reproduction attempt. And so over time, these choices become drivers of a species' life history pattern, and in particular, their ecological breeding strategy. And the critical question in this area is, when do females choose to stockpile nutritive resources versus when to spend them in order to meet their energy budgets? Now, at species at risk of extinction, we're particularly interested in these endogenous signals, which allow females to make the choices important for successful reproduction. And so understanding these signals are a key way in which we can modify and improve our management strategies and promote species survival. And sea turtles are an interesting system for studying energy budgets because of the fact that they have a very high reproductive output. Sea turtles lay 10 to 100 times more eggs per season than any other reptile. And they combine this high reproductive output with a long reproductive migration, and they haul out on the beach to nest as well. Now, these species don't exhibit any parental care. So when we think about the egg, that content of the egg yolk is a particularly important way in which females invest in the success of their offspring. And so when we've actually looked at the effect of nutrition on female reproductive success, what we've seen is that in sea turtles, if environmental variation affects their feeding, their prey species, what we see is that they lay fewer eggs, they lay fewer nests, and they come back less often to breed. And so if we can identify important habitats in which females need access to better nutrition, we could directly influence these important parameters like egg number, which could help in supporting the species survival. And so when we think about each egg a female invests in her offspring, she is investing a certain dollar amount. And so we want to know essentially, when are these females stockpiling their resources and when are they spending them? And so when we look at the classical model for large marine reptiles, we see that sea turtles have always been considered capital breeders. What that means is that they store up energy capital during non-nesting months in order to stockpile nutritive resources. And so they then draw upon this stockpile over the course of the season to spur their reproductive activities. And whatever is left at the end of the season has to spur that female's reproductive migration back to feeding grounds. However, in my species, we're seeing quite a bit of differences in their life history pattern, which indicate that maybe capital breeding, which works well in other sea turtle species, may not work as well in the olive ridley sea turtle. And so, for example, one thing that we've noticed about the olive ridley, one is that they are highly nomadic. And so other species of sea turtle are likely to use common migratory corridors and to have specifically defined areas where they are likely to be found feeding. But not so with this species. In fact, because the species disperses so far, we've actually had difficulty defining those foraging areas. We also know that the life history pattern of the species is a little bit different because they come back to nest every year instead of taking years off. And at the nesting grounds, our species has access to adequate prey. So they don't necessarily have to fast as in other species. And so taking all of this together, I hypothesize that olive ridleys are meeting their energy budgets by feeding at the nesting grounds. And so in order to test this, I spent 3 summers in Costa Rica in which we attempted to capture and recapture our females in different reproductive states. So we went down to Astianal, Costa Rica, and we were able to catch females at the mating period, at which point we would wait for males and females to stop mating, and we would pull them out of the water in order to take blood samples and ultrasound. And then what we would do is we'd tag those females, and we would recapture some of those females again on the nesting beach, as well as sampling other nesting females we found. And so the goal of this study was to look at hormone and lipid profiles over the course of the season to see if we could determine when females were using their stockpiled energy. And so the first question we asked was, are turtles using lipid during nesting, and how much are they using? So we chose for this measurement vitellogenin, which is a huge yolk lipoprotein that is produced by oviparous vertebrates as that nutrient source for the developing embryo, and it becomes the yolk. And so we wanted to know if we could detect this protein changing over the course of the season. And this is an assay that was developed during my master's at Southeast Louisiana University. And so when we take these samples and we look at vitellogenin, what we can see is that vitellogenin remains high throughout the season. So when we look at these data sets, you can see our initial sample at post-mating, and then samples again at nest 1, 2, and 3. And what you can see is that that vitellogenin concentration remains high through the end of nesting season, with about an average of 38 milligrams per mil. And we also did a biological validation and saw that we could not detect it in males, so that was good. Okay. And so if these females are using a lot of lipid, then where are they mobilizing it from? And one thought we had was to use ultrasound in a newer technique in which we can actually measure subcutaneous fat depth with the ultrasound probe, non-invasively. And so essentially what we can do is look at the layers of skin. And so the gray layer you see at the top of the screen is the epidermis. Layer C is skeletal muscle with the shoulder joint highlighted as a marker. And then that black layer in the middle, B, is what we're actually measuring as subcutaneous fat, the anechoic region. And so we take 3 of these samples, average them, and get a fat layer diameter for each turtle. And so we predicted that over the course of the season, if females are mobilizing fat from subcutaneous stores, then we should see a decline in the fat depth from the mating period to the nesting period. And so when we looked at this, we have our post-mating females, nest 1, nest 2, and nest 3, and we can see that there's no significant difference in fat layer depth as we go across the season, which to me indicates that it's not likely that they're mobilizing fat from this region, but of course we know that there are other places they could be mobilizing fat from. So we started to think about another metric we could use, which is ketone bodies. So if we want to know if the liver is mobilizing lipid in order to meet this need, then we can look at BHB over the course of the season, beta hydroxybutyrate, which is an indicator of fasting and increases over 400% in sea turtles over 2 weeks of fasting. And so when we looked at ketone bodies over the course of the season, when we look at our post-mating female, okay, well, before I say that, I should say we expected to see an increase, right, over the course of the season if the females are not eating and they're fasting, they'll continually be mobilizing lipid stores from the liver. And so when we compare our mating individuals to our recaptures, we can see that there's no significant difference. And we actually don't see the increase that we expected, which to me indicates that the females may be feeding over the course of the season, keeping those BHB levels relatively low, or at least she's not needing to mobilize a whole lot from the liver as we go from early to late season. So what I've shown you so far is that vitellogenin remains high throughout the final nesting event, and that subcutaneous fat layer diameter does not decrease. And when we looked at organ lipid stores such as BHB, we also found that those do not increase over the course of the season. And so this leaves me with one final question. Okay. Are females actually feeding at the nesting ground? And luckily, there's a hormone that is a perfect candidate for this. And so in our next phase of research, what we're planning to do is look at ghrelin concentration. And we've done a little bit of this to start with, because ghrelin has been shown to be orexigenic in many vertebrates. We know that it's secreted following stomach emptying, and that it targets the arcuate nucleus in order to stimulate feeding behavior. And so we know that ghrelin also drops during food intake, and so if we can measure ghrelin in our turtles and look at the patterns over the course of the season, I think we can determine whether it's likely or not that females are actively taking in food. And so in our preliminary data, what we've seen, what we expected was to see low ghrelin early in the season and continuously increasing to the end. And that's just not what we're seeing in our individuals. So in our pilot study, we ran a few females from the post-mating group, nest 1 and nest 3. We can see that these numbers are not dramatically shifting. And so when we increase the sample size to look at this, it will be interesting to see if this pattern persists or changes. And so I'm proposing that olive ridleys could be income breeders. And so income breeders rely on active foraging throughout the reproductive season to support reproductive effort. And so when they do this, they are able to maintain their fat layer throughout the course of the season and supplement their needs for energy with active food intake. And so you might be wondering what the importance of this question is. And the answer is that in all endangered species, nesting habitat and breeding habitat are maintained in completely different sets of objectives. And so if we can show that our females need access to nutrition during this feeding period, if they are actively feeding to support their reproduction, then we need a massive overhaul on all of our management plans that we use currently for managing nesting beaches. Because at this time, we do not monitor prey species at nesting beaches, and we don't concern ourselves with supplementing in any way. And so we don't know if females have a need for nutrition at this location or how it could improve until we actually establish a baseline that there is feeding behavior occurring and we need to look at this. And so with that, I'd like to thank all of the many undergraduate students that have helped me over the years, my committee, as well as Dr. McKenzie, who's been a great resource and inspiration to me throughout my career. And I would also like to mention that we imported all of our samples with the appropriate CITES permits. And I would be happy to answer any questions you have. So what we found is that the model is that females nest in a discrete period as soon as they arrive at the nesting grounds. And then they need at least 2 weeks in order to ovulate and prep a clutch. But oftentimes we won't see a mass nesting event called an arrebata, which is where I find all these females, for up to 6 weeks after we capture them at mating. So it's somewhere between 2 to 8 weeks between mating and nesting.
I have a question unrelated to your data from.
Okay.
Off the sound question made me think, is there some knowledge on these turtles about follicular reserves, very reserves?
So there's not a whole lot of data on turtles that are very old. So I would say we know that these females are likely to reproduce throughout their lives, and that they have very dynamic ovaries, which have to facilitate these really high clutch numbers, right? And so as far as ovarian reserve, we've actually talked about trying to look at that with a colleague of ours, Dr. Jacques, who looks at anti-Müllerian hormone, who seems to think that it might be related, that you might be able to correlate that with ovarian reserve. And so we're interested in that. The only problem that makes it hard for us to address that with our data is that we can't get follicle numbers from ultrasound because there's so many and they all look identical. And so we have hundreds of follicles in here, and so it's really hard for us to make any correlations. But, if there was a study where we could actually dissect a female that had washed up on the beach or something like that, that would be something I'd like to do.
Our last trainee speaker today is excuse me, Viviana Garza. She's a student with Dr. Cordoso. The title of her talk is Impact of Pre- and Postnatal Nutrition Extremes on Tonic Secretion of Gonadotropins and Feedback Responsiveness to Estradiol in Sexually Mature Heifers.
Thank you. Can everyone hear me okay? All right, so optimal lifetime productivity in heifers is largely dependent on their ability to reach reproductive maturation, conceive early in their first breeding season, and calve for the first time by approximately 24 months of age. Therefore, understanding the long-term effects of pre- and postnatal nutrition on reproductive functions is crucial in order to develop efficient management strategies geared towards promoting this optimal lifetime productivity. And so there are varying factors that can have impacts on reproduction, one of those being nutritional stress, and this includes starvation and obesity. And so these nutritional perturbations can have varying impacts on reproduction that may occur either pre or postnatally. Developmental changes that occur in response to nutritional extremes prenatally and during the early postnatal period are believed to be imprinted in the genome and in some cases may be manifested only later in life. Our laboratory has previously focused on the effects of nutritional programming during the postnatal juvenile period. However, more recently we have begun to examine the interactive effects of pre- and postnatal nutrition on reproductive processes during adulthood. The overarching aims were to determine whether nutritional extremes during prenatal development interacted with postnatal nutritional extremes during the juvenile period of heifer offspring to impact reproductive phenotype. And so for these experiments, it was hypothesized that nutritional extremes during the second and third trimesters of pregnancy will interact with nutritional extremes during the postnatal juvenile period in order to impact pulsatile secretions of gonadotropins as well as estradiol feedback responsiveness. 108 cows were fed to achieve a targeted body condition score by 6 months of gestation and maintain until calving. However, due to factors such as dystocia, error in pregnancy determination, as well as outliers in heifers that did not reach puberty, the final number of females that were available was 95. And so maternal nutrition consisted of 3 different treatment groups, and those were represented by a low group, which targeted a body condition score of 3 to 3.5, a moderate group, which targeted a body condition score of 5.5 to 6, and a high group, which targeted a body condition score of 7.5 to 8. And so at the beginning of the second trimester, body condition scores and body weight were recorded every 2 weeks until partition, with diet changes made in accordance to changes in body condition and body weight. And so as we can see at time 0, there were no differences between the groups. However, as time progressed on their respective treatments, we can see that the groups begin to diverge and the targeted body condition scores were met by partition. The heifer offspring were then weaned at 3 to 3.5 months of age, where they were then assigned randomly within maternal treatment, and those treatments consisted of a targeted a rate of gain of 1 kilogram per day. And so they remained on these on a common growth diet until they reached puberty. And so once they were placed and at approximately 4 months of age, they 2-week intervals until 8 months of age. And so as we can see at time 0, there were no differences on their respective treatments. A significant difference can be observed as well. With 3 maternal treatment groups and 2 postnatal treatment groups, a 3x2 factorial resulted in 6 different treatment combinations. However, for these experiments, and those included the nutritional extremes, which are represented by the high experiments that indicated the unlikelihood of observing measurable a stable neuroendocrine model that wasn't subject to changes due to estrus, ovulation, or other factors. The heifers were overrectomized upon plastic implants in order to create a physiological baseline concentration of estradiol. The heifers were sampled in a full-fed state, which resulted in a total of 18 females available for these experiments. The first experiment looked at the impacts of pre- and postnatal nutritional extremes on tonic secretion of gonadotropins in sexually mature heifers. And the objectives were to assess the effects of pre- and postnatal nutrition on pulsatile secretion patterns of gonadotropins among the different nutritional treatment groups. All the heifers were previously trained and acclimated to the experimental conditions inside of a large animal sampling facility. And 24 hours prior to the initiation of sampling, indwelling jugular catheters were installed into each heifer and sutured to the skin of the neck, where they were then maintained by flushing with a heparin solution in order to prevent clotting. The following day, the heifers were brought into the large animal sampling facility. They were then placed in stanchions and allowed a 30-minute adjustment period. This was followed by a sampling window of 5.5 hours, utilizing 10-minute interval sampling. Plasma was collected, and serum concentrations of LH and FSH were measured via radioimmunoassay. Individual pulses of gonadotropins were determined as described in previous works, and treatment effects on frequencies and amplitudes of gonadotropins were analyzed by using a one-way ANOVA, and to compare mean concentrations, mixed model analysis for repeated measures was used. Figures from 2 heifers in each nutritional treatment group can be observed on this slide, and these figures are representative of pulsatile patterns of LH. When evaluating LH pulsatility, the moderate-high group tended to have greater amplitude of pulses when compared to the high-high and low-low groups. However, in regards to frequency of pulses and mean concentrations of LH, there were no significant differences observed between the different nutritional treatment groups. When evaluating FSH pulsatility in regards to frequency and amplitude of pulses, as well as mean concentrations of FSH, no significant differences were observed between the different nutritional treatment groups. The next experiment looked at the impacts of pre- and postnatal nutritional extremes on feedback responsiveness to estradiol in sexually mature heifers. So estradiol imposes negative feedback effects at the medial-basal hypothalamic level via candy neurons, and positive feedback effects at the retrochiasmatic area in order to initiate the LH surge. So the objectives for this experiment was to assess negative and positive feedback responses to a pharmacological rise in estradiol. The sampling methods for this experiment were the same as for experiment 1. And for the experimental design, at day 0, CEDARs were inserted and they remained in place for a 5-day period. This was done in order to induce a progesterone priming effect. On day 5, these CEDARs were removed and jugular catheters were inserted and sutured to the skin of the neck. The following day, sampling began with a 36-minute pre-treatment period that utilized 12-minute interval sampling. At time 0, estradiol 17 beta in oil was administered via an intramuscular injection at a dosage of 2.4 micrograms per kilogram. Sampling then continued for an additional 4 hours at 12-minute intervals. The onset of the LH surge was expected to occur as early as 12 hours post-estradiol injection. Therefore, sampling resumed at hour 10 and continued until hour 22 at 30-minute intervals. Sampling continued at hourly intervals until hour 24, and an additional sample was collected at hour 30 in case any of the heifers had a surge later than the expected onset. And so the onset of the LH surge was defined as the first samples in a series that lasted at least 1 hour and rose 2 standard deviations greater than the mean concentrations of LH during the negative feedback period. And for the period of the surge, the area under the curve for each heifer was determined by using a trapezoid rule, and this was then evaluated by using a one-way ANOVA. And when examining the negative feedback period, a repeated measures ANOVA was used. And so we wanted to ensure that all of the heifers had a LH surge, and so a pharmacological dose of exogenous estradiol was administered. And this dose elevated concentrations of estradiol to pharmacological concentrations. When evaluating the negative feedback period of LH, there was no treatment effect or time by treatment interaction. However, a time effect was observed, and so when administering exogenous estradiol, LH suppression was induced during the negative feedback period. When evaluating the negative feedback period for FSH, no treatment effect was observed. A time effect was observed, however, this suppression did not appear to be as dramatic as when observing LH. And the different treatment groups tended to differ at various time points throughout the negative feedback period. So on the left, we can see that exogenous estradiol suppressed mean plasma concentrations of LH, while then stimulating pre-ovulatory type surges of LH. However, when evaluating the area under the curve for the period of the surge, there were no significant differences observed for the different treatment groups. So in conclusion, neither pre nor postnatal nutritional treatments affected pulsatile frequency of the gonadotropins or estradiol feedback responsiveness. And in terms of FSH surges, only 2 heifers displayed FSH surges. And so this data adds to a growing body of evidence that characteristics of adult reproductive phenotype in the bovine female are resilient to significant degrees of nutritional stress imposed during pre- and early postnatal periods, particularly if early gestation is avoided. And so thank you to my advisor, my lab mates, and with that, I will open to any questions. I'm not That's a good question. I'm not sure. I feel like the animal model that we used is a pretty good representation of extremes with a minimum body condition score of 3 and 8. So I'm not sure. I feel like this is pretty good. Yeah, not sure, but that's a good question.
I got it. I think I got it. Okay, now we begin our last faculty presentation, and from Dr. Chinli Lee, who provides some closing comments as well. The title of his talk is New Insights into Uterine Epithelial Development.
Good afternoon, everyone. So today I'm going to talk about some work we recently did about the uterine epithelial integrity. So this is just a Reminder for those who do not work with the mouse, I think we all know the importance of the uterus in the female reproductive system. And this is the mouse uterus, a cross-section with immunofluorescence staining of the smooth muscle actin alpha. So this shows the uterine wall, and the middle is the endometrium. And the green color represents the KRT8 staining, all the epithelial compartment. So you have the luminal epithelium and glandular epithelium. So actually I have a better ways to localize the uterine glandular epithelium. You can use FOXC2, which is a transcription factor that is specifically expressed in the glandular epithelium, not the luminal epithelium. You can see clearly we localize the daytime uterus. So those represent the gland, uterine glands actually. So the FOXC2 is very important. If you delete FOXC2 in the uterus or in the epithelium, what you're going to see, you're going to have the uterus that does not have any glands. So these mice will be infertile. So I just want to give some justifications for why studying the uterine epithelial integrity is important. So there are two examples. First, implantation. We know that implantation is a process where the blastocysts touch the uterine luminal epithelium and invade the uterine stroma. So you have to have a luminal epithelium, integral luminal epithelium, in order for this implantation to occur. Then the next thing is the uterine glands. We know that uterine glands is quite important, functionally important for many reproductive processes in the early pregnancy. For instance, the implantation, desigilization, and embryo development. This has been demonstrated across different species, including the rodents. And one very important factor is leaf. So in the FOXC2 knockout mice, which do not have glands, if you give the leaf, you can partially rescue actually the implantation and desigilization defects, suggesting the importance of leaf in these processes. Postnatal uterine gland development is in the mouse, starts like with a 0, you have nothing. Actually, you don't have any uterine glands. And later at day 5, you see the luminal epithelium start to evaginate, form some bud. And then day 7, you see some invaginations, and those actually go into the stroma. Then at day 15, essentially have a lot of uterine glands, abundant uterine glands. So I always like to compare this with the seeding process, like you grow plants. So the seeds will be the luminal epithelial cells. So those cells have to be good. They have to be proliferative. They have to be migrative, and in good quantity. So for the soil, I refer those to the endometrial stroma. So the stroma has to be receptive. They have to be rich. And also permissive. So recent studies from Dr. Richard Burns' lab actually using the 3D imaging, they provide some new insights in the uterine gland development. So we know that 3D imaging is superior to the 2D imaging because of its enhanced resolution in the depths. So in his study, he demonstrated that uterine glands actually uterine gland development actually undergoes several stages, including the aglandular stage. The same thing like you see in the 2D, you don't see any glands. And then the next stage will be the bud stage. So you clearly see those buds. And then you have teardrop glands, you have elongated glands, and then you have sinus glands, then you have branching morphogenesis, you have all those glands. So the timing of the uterine gland development from this 3D imaging actually correlates very well with findings from the 2D studies. So in our recent study, we demonstrated if you dysregulate TGF-beta signaling, actually you interfere with the uterine gland development. So when you overactive the TGF-beta signaling in the uterus, what you saw the endometrial differentiation was affected, and the formation of the endometrial compartment was altered. This lead to the defects in the gland development. We also call it adenogenic processes. So adenogenesis and uterine gland development are just similar terms. So I'm not going to talk a lot about the TG beta signaling, but just a quick reminder. So this is a very large family of growth factors that play important roles in gene regulation and development. So you have ligands, receptors, and SMAD proteins. This is a signaling paradigm of the TG beta signaling. The ligands bind to the receptors and transduce the signaling through the receptor regular SMADs and then common SMADs and regular gene transcription. So we generated a mouse model using the constitutively active TGBR1 in the uterus. So when we look at the developmental dynamics, what we found that this is day 5. So day 5, then there's actually not a lot of no uterine gland development actually. So that correlates with the previous report. This is the control, this is CA, the similar results. And day 7, you start to see some uterine buds in this control, but not in this CA. And also in day 15, you saw abundant uterine glands formed in the endometrial stroma in the control, but not in the CA. CA, you did have some invaginations, but not a lot of uterine gland development. So this suggests that overactivation of TG-beta signaling interfere with uterine gland development. So now first thing we look at is the seeds, the luminal epithelial cells. So because their proliferation is critical, they need to be proliferative in order for normal uterine glands to form. So we performed double immunofluorescence labeling the Ki67, which is the proliferation marker, and the KRT8, which labels the epithelium. So we performed the quantification. We show that at day 7, in fact, you don't see any differences of the KRT7 positive cells between the control and the CA. And day 21, you even see more in the CA. So this suggests impaired luminal epithelial proliferation is not the result, is not a cause for the defective uterine gland development in this mouse model. So now we look at the soil. The soil has really had a big problem. We show that using the trichrome staining, we show the uterine endometrium is very problematic. They have fibrosis. You have all these blue fibers. We did the collagen 1 staining. We show that there's higher expression of collagen in this uterus. So these fibrotic changes is supported by qPCR showing the upregulation of a number of genes like collagen 1, laminin, and integrins. So I think the dysregulation of TG-beta signaling can cause the fibrotic changes in the uterine endometrium and the formation of the endometrial compartment. So that causes uterine gland defects. So we postulated that if you reduce the number of endometrial cells that expresses transgene or improve the endometrial compartment formation, you would possibly can mitigate this phenotype. So for that reason, we generated a mosaic activation of the TGF-br1 in the endometrium in approximately about half of the complement of the cells. So in that case, we did see the uterine gland reform in these mice, and here is control. Now you see in the CA, you also see these uterine glands, and the uterine endometrial compartment was improved. So our studies really suggest the importance of uterine mesenchymal epithelial interaction in the development of the uterus. We know that uterine epithelial development has many other mechanisms that govern these processes, including the thing I just told you about FOXA2, which is important for formation of uterine glands, and a lot of growth factors, particularly the Wnt pathway signaling. So this has been very well characterized. I'm not going to spend any time to talk about this. So the one important question we ask is, how about epigenetic mechanisms? Because that's a really pretty understood process in uterine development. So if you probably still remember in 1957, Dr. Conrad Waddington brought up this epigenetic landscape concept. This portraits the cell differentiation status during development. So this is a ball rolling from the top of the valley this ball actually represents a cell. So this cell can go down through different developmental trajectories, resulting in different outcomes, and the cell fades. We know epigenetics plays a very important role in development of cancer, but in the uterine development, very little is known about this. So we are very interested in one of regulator called EZH2. It's an enhancer of ZST2, which is the trimethylation of H3K27. So this is the PRC2 complex, representing the polycomb repressive complex. So PRC2 is a very important cell differentiation and proliferation. So the EZH2 can catalyze the H3K27 trimethylation. It plays many different roles. It can also target non-histone proteins and also can so we generate a mouse model with conditional deletion knockout allele of EZH2, 14 and 15 of the cell domain, which is critical too. And we analyzed the specifically in the EZH2 knockout uteri, but not in the controls, and script levels, and show that EZH2 was significantly, but EZH1, which is not affected. So this suggests the specific deletion of EZH2 in our model, and reduced. And immunohistochemistry clearly demonstrates that EZH2. So this is the control. You see the expression of the EZH2 in the. So we generate a clean knockout, almost a very clean knockout, and now we look at fertility of these mice. Fertility, so based on the liter per month and the pups per liter, we can also reproduction. So now we characterize the phenotype of this mice. So we found something really interesting. So in the uterus, I believe most of you can still remember what type of epithelial cells are in the uterus. So we know for the luminal epithelium, they are columnar epithelium. But if you look at the knockout, you start to see some very interesting phenomena. The layer of the cells, or part of the layer of the cells, form under this columnar epithelium. We identify them, we use a. But in general, in the uterus, they do not have any basal cells. And it has been shown that basal cell development can potentially interfere with the fertility, and that can also cause cancer development. So these phenotypes seem to be exaggerated when the mice become old. So at this 2 months, it's 8 months, and you can see epithelial stratification are more intensified. And using the P63, we can clearly identify this layer of cells surrounding the luminal epithelial cells. So this is a layer of cells that positive for P63. So perform the qPCR to further support these findings, and we use a uterine tissue and also uterine epithelia isolated from the uterus. So you can see that all the basal cell markers, KRT5, 14, and MP63 were all upregulated in the tissues and also in the epithelial compartment. So now we ask the question whether the upregulation of the basal-like cells, genes, were due to the reduced H3K27 deposition on the promoters of these genes. So we performed cheap qPCR. So to perform cheap, you need to do the cross-linking, chromatin fragmentation, then you do the immunoprecipitation, purification of the DNA into qPCR. So what we saw that this top panel shows the form of the basal cells, and now, but it's not in the controls. So we see this change of the reduced H3K27 trimethylation in the promoter regions of the basal cell genes. This is the KRT14, this is the MP63. So now we think EDH2 is a guardian for normal uterine integrity, epithelial integrity. So what happens at EDH2 in normal uterine epithelial cells, it epigenetically suppress the basal cell genes to prevent the basal cell phenotype. That's why you see the normal uterine epithelial development. When you lose EDH2, what happens that these basal cell genes will be repressed, leading to the formation of these basal layers. So the question we further ask is, what happened in the cancer cells? So what happens in the cancerous epithelial cells, actually? So when you lose the EDH2, we ask two specific questions. Number one, so will this stratification occur? Number two, if this stratification occur, then what is the potential impact on the tumor development? To answer this question, we produce 4 lines of mice. It's a lot of work. So we have the P10 knockout and the P10 control, the P10 EDH2 double knockout, and P10 EDH2 double knockout and its control. So the rationale behind this experiment is that conditional deletion of P10 can cause endometrial cancer. That's a very well-established cancer model to study endometrial cancer model. And so we essentially generate a double knockout of EDH2 and P10 to see what happens of lost EDH2 in the endometrial cancer cells. So to validate this model, we performed qPCR analysis to look at the transcript levels. In fact, in the P10 knockout mice, you see higher expression of EDH2, but in the double knockout, this was significantly reduced. And P10 expression was also correlated very well with the knockout status of P10 or double knockout. So they both are reduced in the uterus of the knockout mice. So this shows the Western blot showing the protein levels was reduced for EDH2, P10, and also the activation of AKT because of no P10 inhibit AKT. So you can expect to see this activation increased phospho AKT, but total AKT was not reduced, not changed. So we validate the model, then we, I want to make the story short, very long story, make it very short. So we characterized the mice, we found something really interesting. We start to look at the phenotype of the tumor burden. So we found that we measure the uterine weight. This is the uterine weight, this is the controls, 2 group of controls. And if you look at the single knockout of P10, you have higher uterine weight. This is at the age of, this is 3 weeks of age. The double knockout P10 EDH2, you see, in fact, there's a slight reduction of the uterine weight. But this can be changed dramatically in a time-dependent manner. So we look at 9 weeks of age, you can clearly see the enlargement of this uterine uteri of the double knockout. And now this double knockout have much higher uterine weight than the single knockout. So it seems there's a biophysic change of the tumor burden in this mouse model. And very interesting, you look at the survival of these mice, starting from 2 months, then there are some mice that start to die in the double knockout, but not in the, based on previous, there's no death of the single knockout, and the control group remain, all survived. Okay, we think that there might be some proliferation problems in these double mutant mice. Maybe the proliferation is altered. That's caused the reduced uterine weight at an early stage. So in fact, to look at this, we performed a Ki67 staining, and Xinfang in my lab went ahead and did an index of the Ki67. She counted all the cells in, between the control and between the P10 single and double knockout. In fact, we see a slight but significant reduction of the Ki67 positive cells in a double knockout versus a single knockout. So that's really well explained our findings from the early stage that you have a reduced uterine weight or tumor burden. But the question now, the critical question we ask is what happened like at the later stage? Why the tumor burden is increased in the double knockout? So we did a lot of studies, and finally we think there might be some problem with uterine epithelial stratification. So I showed you earlier that in the EDH2 conditional knockout mice, that we see stratification in the uterus. But here in the cancerous epithelium, we look at them again, and actually we identify this epithelial stratification also in the cancerous epithelium. So this is a single knockout of P10, this is P10 and EDH2 double knockout. So you can clearly see some KRT14 positive cells in the double mutant, but in the single knockout, at this 3 weeks of age, you've never seen any cells positive for this. We stand with the MP63, that's also identified these basal-like cells in the double mutant mice. And actually here, the qPCR support these findings by demonstrating the significantly increased the expression of the MPs of this MP63 in the double knockout group. So we look at 1 month of age. We went further. We saw that this maybe give you a better picture of what happened. So this is HE staining. This is a control group. This is a single knockout. This is a double mutant. So clearly you can see that double mutant, the uterus are larger than the single knockout. And more interestingly, if you look at this carefully, so actually this in the middle and the lumen, there's all stuck with a lot of cells and cell debris. We look at using high microscope, we found out a lot of accumulation of neutrophils in the lumen of the uterus. And here the E-cadherin is an epithelial marker showing the epithelium of the single knockout and the double knockout. You can clearly see the difference. So the single knockout have a lot of cancerous epithelium, but in the double knockout, a lot of cancerous epithelium fall off into the lumen. So all those things stuck in the lumen and have a lot of fluid inside and neutrophils as well. And you can, when we do the KRT14, you can see the stratified epithelium. So here shows the stratified epithelium in the double mutant, but clearly not in the single P10 knockout. And this is the high magnification showing the stratified epithelium there, and a lot of epithelium have been like slot into this lumen and potentially creating a source of chronic inflammation that causing the death of these animals. And we stain MP63, you can clearly see a layer of basal-like cells in the double knockout. To better view this process, we use the double immunofluorescence using this is the KRT14 representing the stratified epithelium. Then you have KRT14, KRT8 staining normal, not normal, just epithelium. So actually we see in the double mutant the part of a lot of areas stained with KRT14 representing their stratified epithelium and also mixed with some KRT8 positive cells. So this is the lumen in the middle. You can see a lot of staining of the KRT14 positive cells, suggesting those cells they were debrided into that lumen. So because we saw massive accumulation of neutrophils in the lumen of those uterus, so we start to look at the neutrophil infiltration. So we use a specific marker with the LY6G that specifically identified the neutrophils. And very interestingly, at the beginning of the tumor development, actually you don't see a lot of difference between the single knockout and double knockout. Essentially, you see all those neutrophils accumulated or infiltrated in the cancer epithelium in both genotypes. But at a later stage, things changed. At 9 weeks of age, we see in the single knockout, here, you see still a lot of some neutrophils are present in the cancerous epithelium, but in the double mutants, as I said, a lot of cancer epithelium already slot into the uterine lumen, but we look at the stroma. It's very interesting. You see in the stroma, a lot of neutrophils are presented as well, but in the single knockout, you almost never seen any or dramatic amount of cells in the stroma. They are pretty clean. So this suggests there's a hidden immunoresponse or neutrophil infiltration at a later stage. So what caused that? At this moment, we don't really know what happened to that, but we think that's probably stratification. There's previous studies showing that in the P10 knockout induced endometrial cancer, the hypoxia played very important roles in inducing the neutrophil infiltration. So we think about the histology of the stratified epithelium. Sometimes I thought about that in Dr. Johnson's class, and he talked about those stratified epithelium. I thought about because there's only a single layer of the basal cells that they come into touch with the stroma. So potentially the blood supplies are hard to get into the upper layers. That's why the causing this stratification, it recruit more of the neutrophils or causing the slaughtering of those cells into the lumen. So now we are actively testing that. We are doing the hypoxia assays, but I hope I can show you that a little more later. And we also performed the ELISA studies to show that there's increased neutrophil elasticity in a biophysic way, like what we saw in the tumor burden. So this is the 1 month, this is 9 weeks. So 1 month, you actually see there's a slight reduction actually of the neutrophil elasticity in the serum, but it's not significant. But in the later stage of 9 weeks, you see want to look at other number of cytokines. We want to see whether at a later stage of the tumor development, there will be like cytokine storm in the serum of those mice, and not leading to the death of the mice. So this is just a summary. As I showed you in the normal uterine epithelium, the EDH2 is a guardian. It epigenetically suppresses basal cell gene expression, preventing the basal cell phenotype to make sure the uterus can function normally. And the cancerous epithelium, now we know that we have 2 questions. With the first question, we already know that loss of EDH2 can also cause the uterine effect in the tumor developmental trajectories. So what's the implication of these findings? So currently, people try to target EDH2 to treat cancers, many types of cancers, but very little is known about endometrial cancer. So this could potentially give an alert that when we target EDH2, we always think about the beneficial effect to mitigate the tumor development, but we never think about this treatment might alter the tumor developmental trajectory, leading to more serious consequence. So this is something we are very actively testing out to you in the near future. Actually conducted most of the studies I presented today, and we have several former trainees that are also involved in some of the TG-beta studies. I really want to point out a number of IFRB members who either collaborate on this project or help with mentoring the graduate students that make sure a project can move forward. So I particularly want to mention Kayla Bailey's, Monique Reginkos, they are all IFRB members. Even Ivanov is not a member, but maybe we should ask him to join the membership. Dr. Burhardt, Fuller Baser, Greg Johnson. Also, I want to thank a number of people who provide the mice for us. Dr. Lauren Basolin, David Vincent, they collaborate on the TG-beta receptor project. Dr. Franco DiMeo and John Lydon, they provide the PR pre-mice. And also want to acknowledge the funding sources for this project. Finally, thank everyone for your attention. I really appreciate the opportunity to present this work, and thank you. If you have any questions, please feel free to ask. great question. So we have the antibody for integrin, the good antibody, but we have really never. But we did use the smooth muscle actin. So we use smooth muscle actin. We look at the first couple of images I show you. Actin, smooth muscle actin in the TG. A lot of endometrial cells start to express smooth muscle actin. So I think that suggests there's a differentiation problem of those. But interestingly, at a later stage, endometrial compartment, but you do not see all of the smooth muscle, the structure of the uterus. So that's why later we use a mosaic activation to improve that, to try to alleviate. Thank you for that interesting question. That if you inject the from people, so he look at the uterus is that the progesterone, the luminal epithelial. That's one of the things, but within the uterine gland development. But I think them present a glandular phenotype. Those things could, I think.
So many
Have not seen anything about EDH2 because, have not seen anything about EDH2 in those studies. But there's something really interesting is that in the mouse model, when people dysregulate the Wnt pathway, sometimes they see this stratification phenotype. Also, when you dysregulate the FGFR2, and people see some stratified epithelium occur. So that's very intriguing thing. And we do not know the interaction between the EDH2 and those pathways that like the Wnt or FGF, but I do believe could be a very interesting target but we did some of the studies to analyze the transcript levels. We did not see any connection, direct connection, but that does not exclude the possibility that there's some real interaction maybe occurring at an early developmental period. So I think those things are all the important questions, things that we want to address in the future. I have not seen anything about that. I have not seen any report. But I have not seen actually any reporting delete EDH2 in the digestive tract. I haven't seen those studies, but in the uterus, I think this is the first thing we observed. We report that one in Journal of American Pathology. Yes, thank you very much. We conclude the 14th annual IFRB retreat, and I think now we're going to have a break, and we will come back. Let's see, are we on schedule, Dr. Bard? Yeah, we'll come back at 3:15. Our honorable speaker is Dr. Greg Johnson for the memorial lecture. So thank you all. I think really enjoyed the meeting. I hope you enjoyed the meeting, and see you in a bit. Thank you. It's very interesting. A lot of things we don't understand. Yeah, okay. It's too loud. We don't want our conversation.